Hello, this is Mr. Marks coming to you with a screencast for the cell cycle and mitosis. So we are firmly in Unit 9 now, which is on cells and cell growth, and actually organismal growth as well. And to kind of begin to look at that, we got to look at how cells grow. So if you think back to the meme that was on the worksheet with multiplication and division being the same thing in the biological world, that's kind of what we're going to be talking about today. In order for me to go from being a small child to the full size I am now, I had to grow. But in order to grow, I had to divide my cells, right? I had to multiply the number of cells through cell division. And that's what we're going to talk about today. Specifically, I want to talk about the cell cycle and mitosis. There's a little bit more to this that we'll cover in a later screencast. There should be some guided notes posted on Google Classroom for you to follow along. And with that, let's go ahead and get started. So I guess the first thing to discuss is, why do cells divide in the first place, right? We have a picture here that has one cell dividing into two becoming four, and so on. But why does that happen? Well, in single-celled organisms, this happens as a method of reproduction, and it's typically called asexual reproduction. It produces genetic clones. In multicellular organisms, it's also for growth and development. How you go from a fertilized egg to an adult, you have to divide your cells. Can also be used for repair and replacement. We replace a lot of our cells. I've got a slide at the end that kind of shows the life cycle of various cell types in our bodies. Some organisms, like lizards and sea stars, they can regrow and regenerate lost limbs, and they're all using this process of mitosis that we're going to talk about today. So we'll start off very broad and then we'll get more specific. So kind of the process of cell division requires that cells accomplish two different things. The first, they must copy all of the different components in their cells. In order for one cell to become two cells, kind of like you see in this picture in the bottom right-hand corner, all of the material in the cell needs to be copied so that each new cell has everything it needs to be successful and stay alive. The other thing it needs to do is divide so that those components are evenly distributed in each of the cells, which are the new cells, which are called daughter cells. So the parent cell is the original cell. The daughter cells are the two cells after the cell splits in half. And this alternating growth, copying, and subsequent subdivision, all these different activities in the cell are called the cell cycle. And that's where we're going to kind of start focusing, and then we'll get into the mitosis process a little bit, which is part of the cell cycle. So why copy all the cellular components? Well, again, we kind of touched on that. But what has to be copied before division? Let's think about all of the various components in both plant cells and animal cells. First and foremost, you got to have a copy of the DNA in every cell because you need those instructions to make all the proteins and give out the different orders that the DNA provides for the cell. You have to copy all of the organelles. You have to copy the membrane. You have to copy lots of other small molecules like enzymes that help cells to function in a normal capacity. All of that has to be copied before the cell can actually split in half. So cells actually grow in size before they divide, because you got to fit all that stuff in. Okay, so in order for a cell to divide, the first thing you have to do is make a copy of the DNA. That is something called DNA replication. We haven't spent too much time talking about it, and we're still not going to spend that much time talking about it. If you're really interested in that process, take AP Biology next year or your senior year. You'll get a lot more information there. But in essence, the double-stranded, double helical model of DNA unzips at those hydrogen bonds that hold the nucleotide base pairs together. And then a bunch of enzymes come in and make a copy of the DNA. And when they do that, now within a cell, you actually have a duplicated set of all of the cell's DNA when a cell is going to divide. So the first step is you got to copy that DNA within the nucleus. But now you have twice as much DNA in your cell, and so you got to make it a little bit smaller. And so the second thing that happens is that it coils up tightly, kind of like thread on a spool. If you think about DNA just kind of flowing around in the nucleus, it's going to take up a lot of space and be a little hard to move around. But if you coil it up really tightly, now it becomes much easier to move about, and that's going to be really important for the mitotic process that we'll talk about in a few slides. So that kind of leads to a description of two different types of DNA. It's not really types of DNA, it's the same DNA, two different forms of DNA. So when a cell is not dividing, the DNA is being actively used by the cell. RNA polymerase is going in and reading different parts of different genes to make messenger RNA, which can be translated into amino acids, which will be folded up into proteins. That needs to happen on the day-to-day basis. So when a cell is not undergoing the division process, the DNA is going to be unfolded up, uncoiled up in this uncondensed form, which is called chromatin. And this is the only time, or this is the state that DNA must be in for DNA replication and transcription to occur. Now, when a cell is ready to divide, that DNA has to change its form. It needs to condense into chromosomes. And so the DNA molecules, they wrap around themselves and around these other special proteins called histones in order to take up less space and become a more compact shape. It does a couple things. It makes it easier to move. It also protects the DNA from damage as it's being moved around. All right, so let's look at this DNA, these forms of DNA in just a little bit more detail. All right, so if we start over on the left side of this diagram, most of the time, what you see in the nucleus is this diagram that looks like spaghetti or ramen. Right, you have a bunch of uncondensed chromatin that is being read by the various enzymes within the nucleus so that messenger RNA transcripts can be produced and leave the nuclear pores out into the cytoplasm so that transcription and translation can occur, proteins can be built. When a cell is going to divide, though, it has to condense and coil into the chromosomes that you see over here. And if you look really closely in the middle part of this diagram, what you see here is our double helix, and it gets wrapped around these histone proteins, and there's some other things attached to the DNA molecule as well, but we're not going to go into that in too much detail today. But these histones, the DNA coils around the histones, and then the histones can coil around other histones to make kind of a much chunkier version of the DNA, which then winds itself up even further to make this larger X-shaped chromosome, right? When you see chromosomes in textbooks, I'm sure you've seen them in your time as a student. They always look like the letter X with a few exceptions, and there's a reason for that. Okay, so kind of already said this, the DNA is then contained in these dense structures called chromosomes, and these chromosomes are actually still in the nucleus. And that will change in just a little bit. And again, before a cell divides, it has to duplicate all of this. So all of this process, all of the DNA has to duplicate, and then everything has to condense into these chromosomes that you see here. It's a pretty complicated process. The chromosomes, once they're duplicated, they have identical genes that are joined at the central point called a centromere. So what you're looking at here is actually a duplicated set of DNA. Here is one copy, and here is the second copy. You write the 1 and the 2 above. It's a little easier to see. And then they are conjoined in the middle with this structure called the centromere, basically joins them together. That is the same DNA, but just two different copies of it. And then our DNA coils up for all of our various chromosomes. Humans have 46 of them. And then when the cell divides, these chromosomes split at the centromere, so half of the DNA can go one way, half of the DNA can go the other way. Again, we'll get to a little bit more detail in just a second. If you don't believe me that chromosomes look like the letter X sometimes, or if you use your imagination, here is an actual microscope picture. I believe this is an electron microscope picture of some chromosomes. You can see they're not all perfect X shapes, but this one's pretty good. This one's not bad. This one's pretty good. You can see that chromosome shape that we find in all of our textbooks is actually a real thing. Okay, so we've talked about the DNA a little bit, right? DNA has to be copied so that each daughter cell can have a set of DNA so it can carry out its own processes. This is all part of the cell cycle. This occurs within the cell cycle. So we're going to talk about the cell cycle now. It's the sequence of events from the time a cell is formed until it splits into two daughter cells. The overall cell cycle serves to double the cell's parts, and then a division or a split can occur. There are two overall stages which can be subdivided into little stages. The first stage is kind of this outer bluish ring. This is interphase, and this is where a cell spends the majority of its time. We will subdivide those phases in just a little bit. And then there's the M phase, or the mitotic phase, that represents the other 10% of the cell cycle. So when we talk about mitosis, that fits in that M phase. It really represents a very small fraction of the overall cell cycle. So let's look at what goes on in all of these different parts. Now we're going to go down a level into the phases within interphase. So let's look at all of these different phases in a little bit more detail. Actually, before we do that, let's start off with interphase, just a little bit more detail. So interphase can be subdivided into G1, S, and G2 phases. And you can think of this as the daily living or the metabolic phase of the cell. This is what the cell does regularly. It will move into the mitotic phase, that last 10%, only when a cell needs to divide. And so G1, S, and G2 are all kind of the day-to-day state of a cell. 90% again of the time is spent here for a cell, and it's kind of this alternation between interphase and the mitotic phase as cell division is occurring, where you go from interphase for quite a bit of time, you go through G1, S, G2, then a cell divides. Then you go through interphase, then a cell divides, interphase, divides, and so on and so forth. Again, cell organelles double, DNA replicates, protein synthesis occurs. Each stage ends with a checkpoint to make sure it's actually ready to go to the next stage, because if it's not ready, then the cell can't survive. And so there actually are some fail-safes built into our genetic code, which is pretty cool. All right, let's look at G1. So these are the events that occur during G1. G, think about G standing for growth. So the G phases are all about growth. There's a lot of cell growth. The chromosomes are preparing for replication. There's a duplication of various cellular components. The cell has to increase its supply of overall proteins. It has to increase its supply of other organelles. And the chromosomes, importantly here, are single. At the end of G1, there is a checkpoint or a restriction point, and this is a point that the cell needs to pass in order to continue on. Otherwise, it's actually going to exit the cell cycle and move into a state called G0, where it will never be able to divide. A lot of our cells actually live in the G0 state. But if they pass that checkpoint, then we move into the S phase, or the synthesis phase. This is where DNA replication occurs, where DNA makes a copy of itself. The DNA replication results in the duplication of all of those chromosomes. And so now you take that one chromosome and you've duplicated it. It's connected by a centromere. And these are two sister chromatids. And that's what this term here is. So a sister chromatid is simply two sets of the same piece of DNA that are conjoined at the centromere. Okay, after the S phase, after the DNA has duplicated, now we go into G2. G stands for growth, so this is another growth phase. More growth occurs. More proteins are produced. These ones are going to be really critical to cell division, to the mitosis phase itself. Again, there are more checkpoints before the cell actually gets into the M phase. Excuse me. And once they get there, then the cell can move into that mitotic phase. So under a microscope, and you kind of looked at a bunch of cells in an onion root tip for your first homework assignment in this unit, and you're trying to figure out what different cells, or you were trying to identify cells that look different from each other. And after this lecture, you'll probably be able to go back and identify the actual phase that they belong to. So in a plant cell, G1S, G2, they kind of look like what you see here on the right. You're going to have a nice defined cell wall, and the nucleus is going to be intact, present, and most of them are stained. That's why you see the pinkish color. In an animal cell, it's going to be similar, but a little bit harder to see because you don't have that nice defined cell wall, but you still see the nucleus holding all the DNA inside. When you see something that looks like this, you're looking at interphase. Hard to differentiate if it's G1S or G2 with a regular light microscope. Okay, let's move into the M phase or the mitosis phase. This is when the DNA actually gets divided and separates, and where one cell divides into two daughter cells. So there are quite a few stages to this. We're going to start off in something called the prophase, then we'll move into metaphase, anaphase, wait for it here, telophase in my animation, and then it goes back to interphase and will eventually reach back to prophase as cells continually divide and move through this cell cycle. Again, remember, cells spend about 90% of their time in that interphase stage. These 4 stages of mitosis represent 10% of the overall cell cycle. So let's look at this in just a little bit more detail. It all starts with prophase. A bunch of things happen in prophase. So I know we mentioned the centrioles in the last unit when we were talking about organelles. These are special organelles that are going to be really important in mitosis. And they're going to move to opposite poles, think like magnetic north and south on Earth. Except in this case, we're kind of going east and west, just because that's how most of the textbooks draw it, but it can happen in any direction, really. But if you look here, the centrioles are going to move towards the poles. The mitotic spindle is going to form. I'm going to talk a little bit more about that on the next slide, which has to do with the centrioles. So they do play a super important role in the mitosis process. The DNA will supercoil and condense into those sister chromatids. Again, the S phase has occurred, so everything is duplicated, so we should see X-shaped chromosomes. They're going to become visible under a light microscope because they're getting so condensed. And really importantly, the nuclear membrane is actually going to break down in the nucleus. The nucleus is going to disappear for a while because if a cell is going to divide, all of the organelles need to be equitably distributed, but you also have to make sure that the DNA is equitably distributed. And you have to get rid of the nucleus in order to do that because that DNA has got to move all around the cell for a little while. And so that is what's going to happen. So just to recap prophase, centrioles move towards the poles, the mitotic spindle forms, DNA supercoils and condenses, and that nuclear membrane disappears. The mitotic spindle is a very special structure that actually attaches to the chromosomes and helps to move them around. So it is a web-like structure made up of these things called microtubules, and it is essential for mitosis because it arranges the chromosomes into their correct positions as the cell prepares to divide. If we look at some actual images of mitosis prophase, they look like this. So if you're looking at the plant cell, we'll look here on the right, you can see that that nuclear membrane is kind of starting to disappear here. The chromosomes are condensing and you see what looks like the little Cheetos that we might think of as a mitochondria if we're talking about organelles. Animal cell, you see something similar. Animal cells are just a little bit harder to see than plant cells for some reason in all of the different microscope imagery that I could find. But you can see, again, the DNA is condensing. The nuclear envelope is kind of disappearing. It's hard to tell where it is, but the DNA is still kind of centrally located in a clump. The next phase of mitosis is metaphase. So in metaphase, the spindle fibers from each of the centrioles, they attach to the centromeres on these sister chromatids, these duplicated chromosomes, and they push them into the middle of the cell. So I think metaphase and middle, and you'll be golden, right? During metaphase, all of the chromosomes line up on what's called the metaphase plate right in the middle of the cell. You can think of it like an equator. If we look at pictures of actual metaphase, you can see exactly that is occurring. Now the nuclear envelope is completely gone. The cell is very much taken up by the DNA molecules, at least in these preparations. And you can see that there is a metaphase plate here. I'm going to actually change my color to yellow because it's a little hard to see writing with red on red. You can see the metaphase plate here. You can see the metaphase plate here. You can see the spindle apparatus here. The centriole must be right about here, and here you can see it very clearly on the animal cell. You can see all of those spindle fibers pushing the chromosomes into their correct positions. This is a really cool microscope prep with some fluorescence imagery where different colors correspond to different things. And this is actually a human cell showing the microtubules in green, the different chromosomes in blue, and the kinetochores, which are part of that centromere, in pink. You can see how they're all kind of being lined up. It's just too cool of a picture not to show. Okay, next phase is anaphase, which is now where this spindle apparatus does the rest of its job. So once everything is lined up on that metaphase plate, now things can start to divide. And so the microtubules, you can think of them a little bit like muscle fibers. They're going to contract and they're going to physically pull apart the sister chromatids, and they're going to separate the DNA, half moving to the left and half moving to the right. The chromatids are now going to be referred to again as chromosomes, and they're going to move towards the poles of the cell. You'll also notice that the mitochondria and the chloroplasts and other things are also getting pulled towards the poles, because this is where this division starts to occur. If we look at this in an actual photograph under a microscope, here you can see that anaphase is occurring. These are being pulled. For some reason, I'm drawing lines. Hit the wrong button here. These are being pulled towards the poles. Being pulled towards the poles. And if I can find it, I have a really cool time-lapse video that I'll post on Google Classroom for you guys that shows this kind of occurring, and it looks really bizarre and awesome. The last phase is kind of the opposite of the first phase. So during prophase, the DNA condensed, the mitotic spindle formed, the nuclear envelope disappeared. Well, all of those things are going to happen in reverse during telophase. Once the chromosomes arrive at the poles, the microtubule spindle fibers disappear. New nuclear membranes form around the DNA that has been pulled to each of those poles. The chromosomes are going to decondense back into the usable chromatin where the different enzymes can read it. And now cytokinesis can begin. So telophase looks a little bit different between plants and animals. In this case, you can see that the cells are starting to form a new membrane here. There's still a little bit of connection right here. In a plant cell, one other step has to occur because plants have a cell wall. Plants actually have to build new cell wall between their cells as this telophase is occurring. Okay, so just to kind of recap the different 4 stages of mitosis, and if you're actually using some textbooks, you'll see 5 stages, prophase and metaphase. There's another phase in some books called prometaphase. We'll just stick with the 4 to keep it simple. So prophase, the chromosomes condense, they supercoil. The mitotic spindle forms, the nuclear envelope disappears, looks like this. Metaphase, all of those chromosomes get attached to the spindle apparatus and they get pushed to the middle of the cell. Metaphase, middle, easy way to remember it. They line up on that equatorial plate in the middle of the cell. Then during anaphase, they are physically pulled apart as the spindle fibers contract. And then during telophase, the new nuclear envelopes begin to form around the separated DNA. And the chromosomes will decondense back into chromatin, and the spindle apparatus will begin to deteriorate. It's really not that difficult. Okay, one last step that lives just outside mitosis is something called cytokinesis. And this is that final split where one cell kind of pinches into 2 using some muscular protein fibers. So there's a fiber ring composed of a special protein called actin, which is actually found in your muscles, and it contracts around the center of the cell, and you can actually see that occurring right here in this top right-hand corner. And it's going to pinch the cell into 2 new daughter cells, where each cell will have its own nucleus. So this is going to happen kind of in that middle plate where metaphase occurred. You'll very often see in textbooks, this is called a cleavage furrow for reasons that are probably obvious by looking at this picture, and I'll leave it at that. All right, so let's kind of look at this in an animal and then a plant, and then we'll wrap up our little discussion here. So if we're looking at white fish embryo cells, so these are just simple fish cells, interphase, you're going to see a cell here, and it's got DNA, but it's not condensed, and you can see a very clearly defined nucleus. Prophase, now that nuclear envelope is dissipating, you can't see it very well. You can see the spindle fibers and the spindle apparatus are forming, and that chromosomes are condensing. Metaphase, everything lines up on that middle equatorial metaphase plate. During anaphase, I apologize, it got cut off. I thought I moved everything up far enough. I clearly did not. But during anaphase, the things are going to begin to spread out. Looks like this is another metaphase picture on the bottom left. But during anaphase, things are going to pull apart. And during telophase and cytokinesis, the chromosomes will decondense. They'll form a new nucleus. The cell will pinch into two new cells. And that is how you take one cell and you turn it into two. If we look at the onion root cells, you see basically the same thing, right? Interphase, you have a clear nucleus. Prophase, you don't. Later in prophase, you're going to see more condensed nuclei or condensed chromosomes. In metaphase, everything lines up. Anaphase, they begin to pull apart. Telophase, everything begins to break down. Later in telophase, you start to see the new cell wall forming, but it basically looks like the animal. These slides are just a little bit better prepared. And so if we go back to a diagram like this that was on your first worksheet, you can probably identify the difference between this one and this one and this one and this one, right? I'm not going to tell you for now because we'll probably do that in an actual discussion in our next Zoom class. Okay, last slide. How often do cells actually go through this mitotic process? Well, it depends on the type of cell and the type of organism. So if you are a fly embryo, the cells divide every 8 minutes. If you're bacteria, you're dividing about every 20 minutes on average. That's a huge average because there are millions of different species of bacteria. Yeast, they undergo mitosis when they're activated every 2 hours. Human skin undergoes mitosis every 20 to 24 hours. So we're constantly reproducing our skin, and we reproduce all of our skin, I think, I want to say, I can't remember the exact time frame, but it's like once every 2 weeks we've replaced all of our skin cells. It could be longer than that, though. I can't remember for sure. Your human liver cells, they don't undergo mitosis. They undergo mitosis about once a year. Human eggs don't. They basically take 40 years or more. Human nerves, they never undergo mitosis, which is why once they're formed, they are what they are. And if you have nerve damage, it's very often permanent because you're not making new nerve cells. All right, that is a little bit about mitosis and the mitotic process. Thank you for tuning in. I will see you in the next Zoom call. Goodbye.